My name is Julie Magarian-Blander. I'm an associate professor in the Immunology Institute at the Icahn School of Medicine in New York City at Mount Sinai.
Why aren't you reading?
Just read it. Oh, I don't know where I work. Oh my God. Forget it. Let's cut myself out completely. I don't want to do this. It's not me.
This is not your part.
Okay.
This is the part
This
you
is the part,
memorize.
a particular type of T-cell response that
No,
we
so
have focused
everything
on.
else, just read it.
My name is Julie Magarian-Blander. I'm an associate professor in the Immunology Institute at the Icahn School of Medicine at Mount Sinai in New York City. Our story is really about how things come together at the right place at the right time in order to induce an efficient immune response against infection. The biological problem that we've been working on concerns the question of how T-cell responses against microorganisms are initiated. A particular type of T-cell response that we have focused on involves CD8 T-cells, which are important for killing infected cells. Now, through their T-cell receptor, CD8 T-cells recognize short peptides presented by MHC class I molecules. These peptides are derived from cytoplasmic proteins. When cells are infected with an intracellular microorganism like a virus, their MHC class I molecules now present peptides derived from the virus. CD8 T-cells specific to the microbial peptides can then identify those infected cells and kill them. The difficulty for the immune system is how to get small numbers of CD8 T-cells that have never before encountered their cognate peptide MHC ligand to find the infected cells in the body. To solve that problem, the immune system makes use of specialized cells called dendritic cells, which chew up infected cells and then migrate to the lymph nodes where T-cells are concentrated. There, T-cells recognize their cognate peptide presented by such dendritic cells and get activated, divide, and expand in number. The T-cells then migrate out of the lymph node. Because their numbers have greatly increased, the chances that those specific CD8 T-cells will find the infected cells and kill them are now much improved. It requires that dendritic cells acquire the microbial proteins by phagocytosis of infected cells and present peptides derived from those proteins in their own MHC class I molecules. This means that they now have to present peptides that are not derived from proteins in their cytosol, the normal source of peptides for MHC class I, but rather acquired from an exogenous source and then presented. This process is called cross-presentation, and it is a unique ability of dendritic cells.